We go to the final session today. So final session today is geared towards talk about the drug toxicity, not necessarily in clinical setting. It's really from the translational setting, where from preclinical to the clinical setting. Because currently in the MPRINT base, a lot of work we've been building of knowledge is really focusing on the clinical trial data, PK studies, epidemiology studies, and clinical studies. But we haven't paid a lot of attention to translational angle from preclinical to the clinical setting. So I think, but this is a big deal for the drug safety studies for pediatric and maternal population. So we have three wonderful speakers today. The first is Dr. Laura Aleksunes. Took me a while. She's a professor in cardiac pharmacy in Rutgers University, and she's recently she's a PI, recently funded, NSHD funded a transportal hallucination center, and starting last year September. Her research is largely focused on biochemical mechanisms of toxicity in general. So let's welcome Laura.
Okay, thank you so much. I'm really excited to be able to be here today. As we talked about, we're going to be looking a little bit at transporter proteins. So why are we interested in transporters? So these are going to be essentially entryways or exit ways for chemicals to get in and out of our cells. And in particular, this is for larger molecules, things like pharmaceuticals, toxicants in our environment and in our diet. In order for them to get across, they need to be able to have some assistance to get in and out. Now, one of the things that has emerged in the field of perinatal pharmacology was that there's been a dearth of data, particularly related to human ontogeny of transporters. And so about 10 years ago, NICHD commissioned a pediatric transporter working group to look at the available data that was there at the time. And we had plenty of preclinical data looking at the blood-brain barrier development in rodents, but really didn't have very much when it came to understanding the context of these proteins. You can see that they're expressed across lots of tissues, and through a number of different other groups, we have a lot of data now on adult and aging, but not in the early developmental period. And only recently, with the publication of our recent transporter review, was the placenta added to the working group. So in last year, in 2023, NICHD, NIDA, and the Office of Dietary Supplements at NIH put out a call for the establishment of a Transporter Elucidation Network, or TEN. And so what they did is they funded 4 centers across 6 different universities, you can see represented here on the slide. And the goal of these groups are to elucidate the functions of different transporters we call solute carriers, largely that bring chemicals into cells, and ABC transporters that largely remove chemicals from cells, and focusing on 4 primary tissues: the human placenta, mammary gland, developing intestine, and blood-brain barrier. And so you can look at the 4 different centers that have been funded, and they really nicely represent all of those different tissue areas, but bring complementary expertise and capabilities to be able to use new translational models in order to de-orphanize these transporters, understand novel functions, as well as understand their regulation. So if you look at the San Francisco group at the top, this is individuals that are very skilled in solute carriers. They're going to be using a lot of cryo-EM and in silico docking, along with molecular dynamic simulations, to understand blood-brain barrier development of transporters and understanding how does it dovetail with a lot of the other physiological adaptations that occur during early ontogeny. At the Johns Hopkins group called MIND, they're focusing on mineral transporters, particularly looking at kind of the connection between the developing gut and lactation and what's coming from milk and then being absorbed, and using innovative models such as mammospheres and intestinal organoids. The UW group is looking at both placenta as well as developing fetal and pediatric intestines. They are world's experts in global and targeted proteomics, also coupling this with placental perfusions, PBPK modeling, and organoid systems, particularly for the intestine. I represent the leadership of the Integrated Transporter Elucidation Center that spans not only Rutgers, but also the University of Pennsylvania, Tulane, Rochester, and the University of North Carolina. So we went pretty broad, and that's why we're called InTech. And what we're doing is we're focusing on the human placenta and using a number of integrated approaches, as our name implies, including microphysiological systems, machine learning and AI, epidemiology, birth cohorts, as well as extracellular vesicles to look at transporters in the human placenta. And so this is our team. As I mentioned, we're spanning 5 different universities here, but really have actually had some previous work together that was able to be proof of concept of our ability to integrate these new technologies and approaches to understanding human placental transporters. And so I want to just share one case study. It's not a pharmaceutical, but it's from a previous project that our team had started working on together that illustrates how we can use these complementary approaches in order to understand new placental biology with regard to transport. So I am a toxicologist by training, and part of our research is also in environmental chemicals, supported by NIEHS. And we're particularly interested in this toxic metal cadmium that we can see detectable in upwards of 97% of pregnant people. Now, thankfully, most people don't have alarmingly high concentrations in their body, but there's certainly relationships as we see higher exposures to cadmium. We have seen this in a number of different cohorts be associated with reduced birth weights. Now, cadmium is very interesting. We're exposed to it through our diet, through other occupational exposures. When we think about cadmium-containing batteries, this makes its way into our environment through electronic waste, as well as things like tobacco use. But it's a pretty pervasive chemical, and it has a fairly long half-life in the body. Now, interestingly, and why we were particularly wanted to look at cadmium is that it's attracted to the placenta, where the placenta actually occurs as kind of a sponge or a sink for cadmium, with actually very little getting across to the fetus. And so suggesting that its mechanisms of toxicity are largely restricted to how it interplays with the placenta. And so we're very interested in one particular transporter that came up from an in vitro screen where we were trying to look at resistance mechanisms, things that could protect a cell against cadmium. And one transporter that came up is the BCRP transporter. Now, when we think and look at kind of the villus structure of the placenta, we have those nice finger-like projections that allow for a great high surface area for nutrient absorption, excretion of waste, and uptake of gases. And when we think about xenobiotic or drug transporters, they're largely going to be expressed on those syncytia trophoblasts right at the junction where we have these villi bathed in maternal blood. The BCRP transporters, you can see, stains very nicely shown in brown on these villi. And what I like to equate BCRP is almost like a revolving door. So as chemicals get into the placental trophoblast cell, very quickly BCRP or its friend, P-glycoprotein, will then kick them right back out. So just like a revolving door at the department store, it's going to move chemicals back to the maternal circulation and prevent their accumulation in trophoblasts. And so, as I said, we had a screening study that suggested that BCRP had an interaction, at least in vitro, with cadmium. And so what we wanted to do was use this integrated approach to be able to interrogate this relationship, recognizing that each experimental approach had its own deficiencies and advantages, and trying to see, could we look across different approaches and see some similarity in responses. So this included using in vitro human cytotrophoblast lines and primary cells, using an epidemiological birth cohort, as well as I'll share from our engineering colleagues, our vascularized placenta on a chip. We also did animal studies as well, but we're not going to talk about those today. They are published in the literature. So in our first initial screens using trophoblast cell lines, we were able to genetically knock down the expression of BCRP and kind of make these knockdown cells that you see in red here, where they had about an 80% reduction in the expression of this transporter. And similar to those early screens that we had conducted, we could see that if you were lacking in those trophoblast cells, function of BCRP, we saw higher accumulation of cadmium at each of these concentrations we tested. And likewise, this correlated then with a heightened sensitivity of these cells to having cytotoxicity. So at least in vitro, in a human trophoblasts, we're able to demonstrate that BCRP could, in fact, reduce accumulation of cadmium and protect against toxicity. But wait, there's more. We then wanted to be able to start to translate this into a human birth cohort that my colleague Emily Barrett organized when she was at the University of Rochester and still runs now that she's at Rutgers University. This is a low-risk, medically healthy pregnancy cohort, 270 individuals that were recruited around 6 weeks of pregnancy, and currently the cohort, the children are about 5 years of age. Well, you can see the characteristics in the infographic we have here. Thankfully, the last baby was born in November 2019, so at least for all of our prenatal endpoints, we didn't really have to worry too much about COVID at that point. So in some of our early studies, we've started using quantitative targeted proteomics with my colleagues at the University of North Carolina to be able to profile inter-individual expression of BCRP across these 270 placentas. And then using traditional epidemiological approaches, looking for different associations and predictors, we screened about 15 different maternal and fetal predictors. And then when we started to build our actual model to look at regulation of BCRP, we could see that 2 key drivers that were important in determining expression of BCRP included parity, which favored higher expression of BCRP, but notably a particular reduced functioning variant known as ABCG2, the gene name for BCRP, and you can see the residue here. And this is either if you are heterozygous for this variant or homozygous for this reduced functioning variant, was associated with reduced expression in this cohort. And this actually replicated a previous cohort we had based out of New Jersey as well. So we wanted to ask the question, well, in individuals that had this reduced functioning variant of BCRP, could we see associations between cadmium toxicity outcomes in this cohort? And so just to kind of summarize, the full data set is published in this paper last year. We actually didn't see a relationship between birth weight and cadmium and exposure in this particular birth cohort, possibly because of the sample size. Likewise, our cadmium concentrations were a little lower than maybe we see in other countries and more polluted areas. But what we did see was that in individuals that had the reduced functioning variant, shown in red, and had higher exposure to cadmium, they had lower placental weights, and then this corresponded with higher fetal placental ratio. So putting more pressure on the placenta to make a sufficient amount of baby. So this started to build on our understanding of the relationship between this transport and the placenta and at least some initial birth outcomes. We then wanted to also be able to advance our engineering models and that we're continuing to build on. And my colleague, Dr. Dan Hu at the University of Pennsylvania, has been able to develop a vascularized placenta on a chip. And this is all unpublished work at this point. You can see he has these multi-chamber areas where he is establishing a support system initially for the development of a fetal endothelial capillary system using ECM and fibroblast supportive cells and inserting primary fetal endothelial cells. And then as the cells get seeded with the endothelial cells on the side, over time, within a few days, they can actually develop perfusible vessels here that form this nice vascular network, very similar to the endothelial capillaries found within the human placenta. At the top here, you can see where we start the maternal side of the maternal fetal barrier, where you can see primary trophoblasts in yellow that are going to be forming syncytiotrophoblasts to simulate what we would see in the intact villus. So you can see how the structure looks as the placenta on a chip starts to develop. This is that vascularized component here. You can see the nice vessels that are formed just through self-aggregation of the endothelium. And then we can also kind of see, as we look at kind of the maternal surface as well, and then how these cells will then be able to syncytialize and form multinucleated cells. In the fetal compartment, we can also perfuse blood cells as well, and it will allow us to be able to translate some of the physiology from the human placenta into this vascularized component. When we use this model, we can also simulate toxicity endpoints. In this case, we brought our friend cadmium back. And what we actually saw is that using this vascularized system with microphysiological flow, you actually can see protection. So the cells are actually more resilient compared to a static condition or a transwell condition. The trophoblasts are a bit more robust, both on the fetal side and the maternal side. And so we see less cytotoxicity from cadmium. However, we still can use concentrations that will obtain a signal. And so what Dan has also done in his team, Poria, they have also added another cell type. So we keep adding in as many different primary cells as we can. And this included neutrophils. And so we wanted to look at the relationship between trophoblast damage and recruitment of neutrophils. And so in this case, not only did they use trophoblasts that were essentially wild type, but also brought in some that were lacking or had reduced expression of our friend BCRP. Again, they saw greater toxicity to the endothelial cells and the vasculature for those lacking BCRP. And as you can see in green, even greater recruitment of neutrophils. So as I conclude, I'm just highlighting again that we're using this integrated approach. Now we'll be applying not only looking at pharmaceuticals, but a number of dietary supplements and nutrients as well by coupling our proteomics in a birth cohort. I didn't get to talk about our cheminformatics and machine learning today, but that is another key component at Tulane University, and our vascularized placenta on a chip in order to deorphanize placental transporters, evaluate maternal fetal transfer, and identify novel regulators of placental transporters using genetics and other endpoints. With that, I'll turn it back to Dr. Lee. I think that's all I had. I'll use the other one. And that's fine. Thank you.
Okay. All right. Our next speaker is Dr. Gilbert Burckart from FDA. He's Associate Director of Pediatrics, and he's a world-known expert in the pediatric pharmacology, served as a president of American College of Clinical Pharmacology for a number of years. So he's talking about toxicity for pediatric patients.
Okay, thank you, Lang, and good afternoon. Thank you all for sticking around and not heading out early to happy hour. Glad to see you here. I was very happy to see them have a session on safety. You know, I've always felt since I've been at FDA that safety was job one. You know, so we really have to focus on safety. Lang says the other thing I have to focus on is finishing quickly. But I will go through my slides quickly, but I think you have copies of them, so that'll be no problem. I know this is a group that's primarily oriented toward maternal health, but all the things I'm going to talk about in terms of secondary targets for drugs, in terms of doing modeling with things like quantitative systems pharmacology, QSP, are all very applicable to maternal health and getting Emma a grant, right? And very applicable to maternal health and fetal pharmacology. And if you're interested in fetal pharmacology, we had a workshop on that about three years ago. You can Google that. You can find it. I think all the recordings even of the fetal pharmacology workshop are still on the web. So let's go. Okay. So safety has always been an orientation of ours, but it kind of got brought to the top again by ICH 11. So there's a new guidance out that from ICH, the FDA published it in 2021. And part of our part of the reason this stimulated me was we've done pediatric extrapolation for a number of years, but we've always done it related to only efficacy. And so with ICHE 11, it says you can not only extrapolate efficacy, but you can also extrapolate safety. And that's there's a risk associated with that, and one that I thought we had to address. And they didn't mention secondary targets in this ICHE 11, but I think secondary targets, particularly related to developmental safety, are particularly important and should be a critical part of a pediatric development plan. So these targets should be specific and they should be quantitative, and we'll talk about quantitative. Okay, I'm going to go over some pediatric studies, talk about guidances and kind of what some of our programs that we have going on now. Okay, in 2017, we published this on neonates. We went back and Jian Wang found 40 different drugs that had included at least three neonates. And 20 of the 40 drugs had serious adverse events associated with them. There were 9 deaths also associated with these 40 drugs. And the conclusion that I've quoted out of the article here is that traditional means for collecting safety in drug development need to be adjusted, particularly for the neonatal population. And preclinical studies are useful for the identification of unexpected off-target drug effects. So preclinical is also one of our focuses here. This was a study we published in 2019. It just basically says that the adverse effects that you see in adults are significantly different than the adverse effects that you'll see in pediatric patients. And so this is on the left here, you see a heat map, and you see all the adverse effects lined up against the antipsychotic and antidepressant drugs. And basically, particularly at sedation, you see a bright red line there. And on the right, you see basically the drug as related to sedation and the risk differences. And sedation is a particular problem for this class of drugs, and that's a big problem for pediatric patients, as you can imagine. So the takeaway points were the adverse effects are different, and probably the developmental biology behind those adverse effects are also very different in pediatric patients than they are in adults. So then we published this in 2021, I believe, this was using labeling. This was actually coming from a workshop we had on pediatric dosing. And we took particular care to look at, we used the typical classical type A, type B, type C, type D, and type E adverse effects. And so we took a very close look at particularly B, C, and D, because those were not the on-target effects you would expect. And basically, takeaway was that a number of drugs had a different increase, had an increased incidence of adverse effects in pediatric patients. This wasn't just all the CNS agents like the antipsychotics and depressants, and this varied across a number of therapeutic areas. And so there were also 15 examples of drugs that failed to get a pediatric indication because of safety reasons. So the question is, can we do a better job than we're doing now of assessing safety, particularly in the preclinical space? And so what do we usually do? Well, one of the things that sponsors do is to screen for secondary targets during drug development. Why? Well, they don't want their drug to get into phase 3, and then all of a sudden you're seeing a huge number of off-target serious adverse effects. And so they do a screening with receptors, and this can be as many as 700 different off-target receptors that they may screen for. Right at the present time, there's no pediatric-specific secondary targets that I'm aware of. We also do juvenile animal studies when we request that of sponsors, and that's now under ICH S11 guidance. Okay, so this is basically from Becca Raz, who's in the Office of Clinical Pharmacology. Let me go back. And so basically, this is looking at the secondary targets that sponsors screened. It shows here 480 sponsors screened over 1,000 different INDs and screened these secondary targets. And you can see that's almost every therapeutic area is covered here with secondary targets. Now, this is important to us at FDA because we're stressing now novel alternative methods to using animal studies, or NAMs. And we're not trying to get rid of animal studies entirely, but we think they should be, they actually should be, we should add novel alternative methods as a way of screening these. And this is just not FDA and NIH. This is, I just took this as a screenshot from the agenda of a workshop that was in October. And it's just FDA. It was FDA, NIH, NSF, NASA, NIST, EPA. So all the governmental agencies now are looking at novel alternative methods, or NAMs, for to animal studies. So basically looking at in vitro studies. So for juvenile animal studies, we have two guidances available for use. The S11 is the newest one. And, in 2011, an early phase of pediatric drug development, Melissa Tassinari from Division of Pediatric Maternal Health said that, well, based upon the evidence at that time, they thought there was a trend toward more directed, specifically designed studies in single species for juvenile animal studies. In fact, that hasn't worked out. Hasn't worked out at all. And if you look at this publication from our cancer group, Rick Pazdur and Greg Riemann, in 2016, they had some real concerns about juvenile animal studies and whether they could address a specific safety concern, not just the safety issues that were seen in adults, but safety issues which you would be specifically looking for in pediatric patients. Well, how do you know that? And that's what we're going to talk about. So this is looking back. We collected 165 labels with juvenile animal studies in them. And these are the things on the right-hand side they were looking for: bone growth, sexual maturation, neurodevelopmental. Those were all very high on the list. And only about, actually, a very small percentage of them had actually had an impact. Only 10% or so had an impact on the actual labeling. So we're doing lots of juvenile animal studies, but we're not getting much bang for the buck. So how can quantitative approaches like physiologically based pharmacokinetics? I think most people in here know about drug concentrations, pharmacokinetics, pharmacokinetic modeling. And how can that help us? Well, everything is quantitative. Everything in biology is quantitative, right? And so this is an example here. Oh, let me get my thing out. Oh, I got to finish in 4 minutes. So basically, everything in biology is quantitative. Everything. And so basically, this is I came from Bayer, a study from Bayer. Basically, they were using deltamethrin, which is a classic neurotoxin. And they were looking at the concentrations from an in vitro test that was meant to supplant animal studies, and seeing that when they use physiologically based pharmacokinetics, they actually found that fetal brain concentrations were far below what they were using on the in vitro. So in other words, we have to couple these new testing methods with what we know about concentrations from a quantitative standpoint, using things like physiologically based pharmacokinetics. And so this is a weight of evidence, which is from that ICH S11, that the FDA accepts now as a guidance, and looking at weight of evidence. And so several of these things are related to weight of evidence, like effects on developing organ system. Pharmacologic target has a role in organ development, or selectivity and specificity of the pharmaceutical. How do you know those things? How do you know those things? Well, you can take a guess and put it on the scale here, but you know those things through developmental biology. That's how you figure it out. And so developmental biology, or systems biology, and then systems pharmacology, and then you add a quantitative piece to that, you get quantitative systems pharmacology, which is basically a very advanced way of modeling, like drug concentration modeling. So we're seeing a lot more of these at FDA. Right now, we've had 400 different submissions to the FDA using QSP. So this is not something new. Companies are doing this and with their drug. They want to understand their drug better, so they're using QSP, and they're submitting this information to FDA. And so we're doing a QSP. This is a colleague of mine, Galar Albawardi, is doing a QSP study now that was sponsored by the Perinatal Pediatric Health Center of Excellence at FDA. And we're looking at bone growth using a QSP model and developmental biology. And basically these biomarkers turn into receptors. And so basically, if we understand the biomarkers behind the biology of bone development, then we also understand the receptors. And if we understand the receptors, then we can use them as secondary targets. So, who does these secondary targets? Well, I told you all the companies do. There's even this commercial program now by Certara, which is called secondary intelligence. And so basically on the left-hand side, you see the receptors. On the right-hand side, they've even kind of gauged this into a stoplight, red, green, yellow, and as to whether these are very important in terms of the new drug binding to a secondary target that's going to cause an adverse effect or an effect upon developmental biology in the pregnant woman or the fetus or the pediatric patient. Okay, so we need to put this all together. And basically not only using our animal studies, our juvenile animal studies, they're still valuable to us, but using QSP and secondary biological targets, using things like prior class experience. We had a speaker at FDA, Nicholas Tatanetti from Cedars-Sinai, and he's using artificial intelligence and machine learning to look at our experience with other drugs before and whether that affects the ontogeny of developmental systems in pediatrics. And so quantitative systems pharmacology, quantitative structure activity relationships, all this can be put together preclinical to hopefully do a better job. So part of this all needs to be part of the plan. We need some new approaches to safety studies in pediatric patients and obviously in maternal health. And developmental biology will help us do that, to incorporate that with quantitative studies and to do a better job of doing a comprehensive safety assessment in all of these patients. Thank you very much.
Thank you. Yeah. Good job. The final speaker today is Dr. Lynne Yao from FDA. Again, she is the Director of Division of Pediatrics and Maternal Health, and she actually, her operation oversees the drug studies in peds and pregnant women and women lactation.
Thanks, Long. Okay, we made it. Well, you got about 12 minutes, I think, is all I'm going to need. So I really appreciate the chance to come and speak at the end of a very busy first day. My talk, I would say, is science light. You've heard a lot of really interesting and very, I think, important science. But ultimately, at the end of the day, all the stuff that you're doing has to be translated and turned into actionable recommendations that we can give to prescribers and patients. And I'm just going to give you a little taste of kind of what we do and why I think what's going on today, what's gone on in the 2 lectures that were given earlier, are important in helping us to communicate information to patients and prescribers. So let's see. All right. Just want to thank 3 people in my division, Dr. Johnson, Dr. DiNatale, and Dr. Shaheen, who helped me with some of the slides I have for this talk. So all of you know this, not going to go over it. There are 2 bullets that I want to highlight. The first bullet is bullet number 4. Many people don't recognize this or understand this. That when a drug is approved in an adult population, that includes pregnant individuals, okay? Unless it's a pregnancy-related condition. So if you're developing a drug for preeclampsia, obviously you're going to be studying pregnant individuals primarily. But once a drug is approved for an adult population, it's not like pregnant individuals are not adults. Most of them are. And I could, you could argue, and I would agree, that there's a small subset of that pregnant population that really are post-pubertal, and are in that adolescent population. The other thing that I think is really important to understand is that most drugs, new drugs that are approved, they're approved with only non-clinical reproductive toxicity information, which means that there's little to no human safety data in pregnancy that supports the approval, even though I just told you that those drugs are approved for pregnant individuals. So I'm going to close these. So a little bit about labeling, and I don't want to get too wonky. And I know I had to cover up my pointy head as a regulator with my hair today. So I just wanted to explain why we do what we do, and why we did what we did to change prescription product labeling. This is something called the pregnancy lactation labeling rule. I think most of you have experienced the confusion related to PLLR implementation. This goes back almost 10 years now. We started the implementation back in 2015. So the reason we took away those A, B, C, D, X letters was because we heard from a lot of people. We did a lot of outreach, a lot of special roundtables, public meetings. And what we understood was that the letter system was overly simplistic and it was misinterpreted. Now, a lot of you who are out there practicing, we heard from you after they were removed and said, well, this is a problem. This is a problem because you replace it with something that I can't use in the clinic, you know, and we recognized that very quickly after we started implementing the rule. We took away the letters because we said, okay, it's kind of misleading. But what we were supposed to replace it with were risk summaries. And those summaries were supposed to include data. Okay. And guess what? We didn't have much of it. And so what we realized was that we needed to do a better job in populating the labeling with human data. And unfortunately, we didn't have a lot of it. So I do want to point out that, you know, you think that, what the heck? FDA, you wrote this labeling and it's like a paragraph long and I don't even know what you're saying. So I do want to say that it is a collaborative process across across disciplines within the review division that have a lot of experience. So there's clinical pharmacology, there's toxicology, pharmacology, toxicology, there's clinical, there's statistics, there's epidemiology. And we all come together and try and figure out what is the best way to label. And sometimes we get it better and sometimes we don't. And actually, we would always like to hear from you in case there are some, you think data that are missing or we didn't communicate it in a very clear way. So again, it's integrated and we hope to provide you with a summary of the potential risks and recommendations for risk mitigation. All right, so good example. I just wanted to show it to you. So this resmetirom, so also known as Resdifra, just approved in March. This is the first drug approved for non-alcoholic steatohepatitis, NASH or MASH. It's now called metabolic associated steatohepatitis. So a big problem now with obesity, type 2 diabetes, and this drug was just approved. So this is a new molecular entity, a brand new drug for a new, brand new first treatment available. This is section 8.1. This is the pregnancy information. And what you can see here, I highlighted in yellow, is no data. Now you can read all the rest of it and it talks about the animal studies and then this required background statement that appears in every labeling because the rule said we had to put it. Okay. And then there's a pregnancy registry. So that's another point that I think is important. But anyway, this is commonly the occurrence for a new drug that first sentence is we don't have any information. Now, if you go to the animal data section, which this is longer than the risk summary, and this animal data goes through all sorts of stuff about AUC and multiples of human exposure. And at the end of it, I'm not sure what you're supposed to make of it. But this is all the data we have, so we give it to you. And again, I'm not defending, well, I am defending what we do, but I'm also recognizing that at the time of the approval, of initial approval for most new drugs, we don't have much that will help you in your clinics. So we do rely on post-marketing sources. After a drug is approved, we rely heavily on studies that we can do that will tell us about the drug and its safe use during pregnancy. And guess what's in here? What's bullet number 2? Real world data. So if I'm going to take and all that effort that you talked in that first session about real world data, this has real and important application to how we collect right now safety information in a pregnant population. And the better data we have in real world data sources, registries, you heard about, EHR, claims data, case control studies, all of this real world data, we are actively requiring sponsors to collect, to review, and for us then to make hopefully better recommendations about the safe use of the drug during pregnancy. And then of course, case reports and series, those are always things that we are, I would say, that looks like the smoking gun. It doesn't necessarily tell us, we wouldn't necessarily label with a single case report, but that might help us to look in those real world data sources for more information. So this is my sort of maybe plea to all of you in the room. So, and what's this supposed to do is sort of give you a graphical presentation or illustrate how much data we get in pregnancy, pre-marketing, at the time of drug approval, and post-marketing. So what we usually get at the time of approval and what we ask sponsors that they must do are developmental and repro-tox studies. Those allow us to conduct the clinical trials in pregnant individuals. And a lot of times those studies don't get done till too late in drug development. And so even if you were interested in enrolling a pregnant individual into your phase 3 trial, if those reproductive studies aren't completed, then you really can't. And I don't know how many of you are in here from industry. I think few. But I will say that sometimes that's the strategy, right? Is to, they're focused on the population that they think are going to use it. They're focused on non-pregnant adults. And so they delay oftentimes the repro-tox studies too late. And then even if there was an opportunity to enroll pregnant individuals, we can't do it because we don't have that animal toxicology data. We do sometimes get pregnant individuals who become pregnant during the clinical trial, and we could collect information on them. But a lot of times what happens is they got pregnant, they left the trial, we didn't collect any follow-up. Generally, as I mentioned, little to no data in pregnant individual, or little to no pregnant individuals enrolled, and also, unfortunately, little to no PK data in pregnant individuals. So this is the other side of it. Remember, I said that when a drug is approved in adults, it's approved for pregnant adults too. But really, are we doing a great job if we don't know if the dose that we're using during pregnancy is actually a dose that is safe and effective? So if you need to make dose adjustments during pregnancy and, there's a change in it's a water-soluble drug and we expect it to be cleared predominantly renally, then during the 3rd trimester, you might expect that dose is going to need to be increased. We don't get that information pre-approval, and we don't get the safety information. So those are the two big whammies. We don't get safety and we don't get dosing. So it's left for post-marketing, and I already mentioned we can get case reports, real-world data, et cetera, and PK studies. So right now, most of the safety data we collect right now in human patients is in the post-marketing setting. This is what I'd like to see, and this is what I hope we can work towards through MPRINT and through collaboration, that we improve data collection in pregnancies that occur inadvertently. Now, our guidance does say that if you identify a pregnant patient in a clinical trial, you can re-consent them. Once they're re-consented, you can do all sorts of things like continue them in the trial, get follow-up. If get follow-up on the infant, you can do PK studies on those or collect PK information on those pregnant individuals. So we'd like to see that a more streamlined process for continuing those patients who become pregnant in a clinical trial. We can increase opportunities for enrollment of pregnant individuals in clinical trials, and we can increase the availability of PK data in pregnant individuals. Now, keep in mind that you don't have to enroll a pregnant patient in phase 3. You could say, I want to do a short PK trial. I know these women are going to consent to take the drug and they're pregnant. Why not do a small PK trial as part of development to collect that dosing information? And then we'd like to increase the quality of post-marketing data collection. Now, how do I tie that all in together with what we just heard from our first two speakers, Lauren and Gil? Well, human data are important and we're not there yet. But if we can refine the safety concerns that we have, if we can use the QSP modeling, if we can look at those toxicology markers that we know are important in the transfer of drugs into or across the placenta, then we can target those drugs that we think are most likely to have an effect. And those are the drugs, those are the situations in which we really need to push to get the information before approval, right? So what you've heard, I think, ties together nicely with the end result. And I'm just here to say thank you for all the work that you're doing. I think MPRINT activities have been hugely impactful already. And I'm looking forward to the rest of the meeting today or in the next couple of days to hear what's next for MPRINT. Thanks very much.
All right, so maybe Gil and Laura, do you want to come up? And I think we can take some questions. All right, go ahead.
Thank you. All the speakers, very interesting. I just wanted to emphasize one of the successes of the BPCA program in pediatrics has been relabeling based on opportunistic studies. And you mentioned them, but you didn't use the term opportunistic. But there are a couple of them. I mean, not only are pregnant women going to get many drugs, they get these drugs routinely. And I'm not quite sure why there isn't more requirement or request or even emphasis in the MPRINT on doing more opportunistic studies, even if it's just PK, because that's good. And then the other very important thing, since we're discussing safety, is the other opportunistic thing is many pregnant women overdose on drugs. And although the dose is often useless, you can get PK data. There are samples, even if you just use waste samples, but I don't know anybody who's doing it. So I'd make a plea for that.
Thank you. If I could just make one comment on that. So fully support what you're saying. And actually, I didn't say it because I thought I'm already the last speaker of the day, and this would maybe even make my presence on the stage even more questionable, but I'm a pediatric nephrologist by training. And so most of my work in the first few years in my role was really to get that BPCA NIH program coordinated. But really now in the last several years, I've focused a lot more attention on the maternal health side because we're farther behind in maternal health, sadly to say, than we are in pediatrics. We're like, pediatrics is probably 20 years ahead of where we are in maternal health. So to your point, we've done a good job on the BPCA side for pediatrics. And I do think there's huge opportunity to improve what we're doing on the maternal health side in using BPCA. The CUDDLE study, which is also ongoing, which, is a terrific a program under NICHD, run by the PTN. They're looking at scavenged or opportunistic samples to collect information on lactation of priority drugs. And I think that the same thing could be said for getting better dosing information on women who are taking the drugs and they're pregnant. So I fully take your point.
Yeah, so I'm going to give a comment and then a question, I think maybe to Lang and or Lauren. Today we've heard about a lot of different approaches and a lot of opportunistic PK study, real world data, microphysiological models, and different non-human based non-clinical models that go beyond animals. And certainly, I think there are a lot of different things that we are funding at the NICHD and NIH, like we talked about in the clinical world, right? Mapping on CDEs, mapping ontologies together, et cetera. It seems, especially to the points that Gil was bringing up in terms of secondary pharmacology, then getting to QSP, and then getting to advanced pharmacometric models that can get to safety prediction, right? And so some of what the question I will get to in a second is, we can't do everything simultaneously just because of resources. So maybe the first part of the question is to Lang in terms of knowledge base, and then Lauren in terms of pharmacometrics. How can we use ontologies to map what the MPRINT hub is doing to then start to bring in other type of data so that we can get improved QSP and pharmacometric models to get to, especially safety predictions. I think that's the thing that scares people more than missing efficacy.
Aaron, wonderful question. I think this is the things I've been thinking about during the past few months as well. Right now, I think that the main gap we have in the knowledge base is really we don't have enough animal model data, in vitro data at all. So I think just trying to get a stage where you engage with clinical translational prediction toxicity, we're not there yet. But a lot of data in the literature, so we basically can do the similar data creation process and trying to kind of get more data from animal models and in vitro studies maybe, but focusing on pediatric and pregnant women, pregnant type of model first. I think that's going to have long way. That is to me is a big knowledge gap in the knowledge base right now.
Yeah, Okay, sorry about that. I think, there is a place, it's going to be difficult for all the existing drugs, but as we were talking about, how can we use these NAMs, especially for new drugs coming online? And a number of the drug companies, especially their DART groups that do the preclinical safety studies, they are looking at a lot of these new NAMs like microphysiological systems and also embracing things like machine learning. So I think that's where if we have some of the case examples like the deltamethrin in vitro study that was mentioned, that you can then feed in known chemicals, you can do predictions that are based on structure. You can use some of the preclinical data, knowing that, the physiology of the placenta is going to be quite different. But if you kind of use surrogate approaches like placentas on a chip or other types of screening assays with the existing data that are out there, then I think you can start to improve that predictions before we can have, maybe, RCTs being done in pregnant individuals and stuff. So I think we still have to do a lot of validation, using historical data before we start really kind of turning it to the forward facing for prediction. So still a lot of work to do.
Thanks for the presentation. Really exciting, and it's good to know all this work and the potential changes in the future. I think just I don't want to make it too long, but if the goal is ultimately to reach to a place where we can study medications in pregnant women in an easier way, whether it's new drug or repurposed drugs, I think the way that it's been done right now, and then some of the requirements and hurdles are pretty extensive. And I was really surprised, and I'm happy actually, from Dr. Burckart's presentation, the juvenile neurotoxic studies and how they inform us little, if you want, about the safety of these medications to a certain degree at a time when they're required to be done on a lot of drugs. That's one thing. So why are we doing it? We get some data from them, but are they useful? Because the doses, when you study these medications, you study them at a dose that are really toxic to the mom. And yet, trying to prove that a toxic dose can cause harm in the fetus, which we all know. So the benefit of doing these neurotox studies at the doses, at the way they're designed to be done, is probably limited, but we still do them, and they cost a lot of money to do. For academic researchers, they don't have the money to do it. So that's limiting some and limiting some work, a lot of work that's probably in this area. And I would just love to hear your feedback about the need for these studies and whether, you mentioned other examples to be replaced, but even if we don't get to that stage right now, because not everybody can do these experiments, what is the benefit in doing studies that we know that they're limited value and they're expensive and they don't actually answer the questions, not just from them, but even animal models in general, they don't provide safety data, especially when you have some clinical human data that could be used. So thank you.
Well, it's NIH that's leading the NAMs, the novel alternative methods. Realistically, they're the ones that organize that workshop, and they have money. We don't have money for grants, but they do. And so I don't know, Aaron or somebody can comment on this, but I'd be very surprised if there aren't some RFPs out there or going to be RFPs out there related to the novel alternative methods. And that is if they're applied either to maternal health or pediatrics, and I think that's an early step. I mean, it's nice that the sponsors are already set up for doing modeling. You know, they have a pool of 100 pharmacometricians just sitting there. They would much rather pay them and do the modeling than they would to conduct a clinical trial for hundreds of millions of dollars. And so, so if you think about pharmacokinetic modeling, well, it's 20 years old, maybe. I mean, in terms of realistically being used in drug development. And so, so we're okay. I think it's the ball is rolling. We're getting applications already with QSP in them. So so I think we just need to continue on that path. But from what you said in terms of from an understanding of the biology and the science and whatever, that needs to be funded by funding agencies, which are in the government. There are a number of them. Not FDA.
Not just about finding the money, but ultimately, if you don't think they're useful enough, why are we spending money on them?
We're up here talking about it, and I think FDA's done a nice job of kind of leading and being on the cutting edge for lots of things. Go to the FDA website, you'll find out about machine learning and AI. And how much of that is being used in drug development now? Well, it's being used cautiously, but I think FDA is very forward thinking. So there's no problem with submitting that information and having those discussions with FDA review groups.
Yeah, could I just add a brief comment on that? So, like everything, I think FDA sort of learns as we go too, and I do think that. We have been in a period for a long time, and I thank the folks in this room who've been part of PREGLAC, who've been part of efforts to advance how we collect data in maternal health, because the labeling I showed you before we changed it. It used to be, I forget what it was, pregnancy, labor, and delivery. That pregnancy section, the clinicians didn't even write it. The only thing that was in that section and before PLLR was the non-clinical people who put the animal toxicology information in there. Clinical people didn't even write that section. So it is, we hear what you're saying, and we recognize that, not just to do a DART study. How much is that going to cost? How much information is that going to really give us? If we already know that there's information in humans is an important question. And that S11 guidance that talks about the weight of evidence. That's a kind of thing that I think I wish I could see, start being used on the pregnancy side. And, E21, which is another ICH guidance. I know there's a lot of numbers here, but E21 is pregnant individuals in clinical trials. And I hope that there is some information and movement there to exactly your point, which is let's think about the totality of evidence. Let's think about what we're really going to get. And then if we don't need to do that animal study, or if we can get the information collected, do something that's quicker, faster, and then get those patients, those human patients enrolled in the clinical trials is what we want, right? So totally hear what you're saying. And I do know that many of you in the audience have been unhappy with advice that FDA has given you. I will just acknowledge that. But again, we are working to try and move this forward for everybody. So,
full disclosure, I'm a modeler. So I look at this from a modeling perspective. And I think there's a parallel set of activities that aren't necessarily being fully engaged in this kind of problem. I'm working with groups on digital twins. And the digital twin approach has been primarily focused on disease models, but I'm pushing them to look at normal development in women's health, and then obviously pregnancy, and even looking at menopause and perimenopause as a means for trying to provide a base for iterative modeling to incorporate the data to continue to refine that. But I think there's a lot of potential for bringing those approaches and those activities into this domain, given the kinds of challenges that you've addressed.
Quick question for Lynn. So Lynn, you talked about the animal part of the labeling right now as being sometimes confusing and sometimes not informative and needs translation. So how good do these alternative modeling analysis have to be before you can consider adding those to the label?
Jeff always asks good questions, and most of the time I don't have good answers. I guess the honest answer is I don't know, but we would be, very interested to have proposals and say, look, right, we think that here's a great model, and we think that, what we already have collected in terms of other molecules or whatever support. And I also think that the goal is not, real world evidence, digital twins, QSP, PBPK, right? All of that is great to get us to the point where I think what we still really need are human data, right? I don't think we need tons of human data, but we need to confirm those findings with some limited human data. And that's, I think, we just need to get there, quicker. I think your idea is good because it'll get us there quicker, and then allow us to enroll pregnant individuals sooner, hopefully before approval. So all of these things, I think, would be the goal to get there more efficiently and hopefully faster.
So FDA works by precedent, except when there's new science. And so when new science gets put into guidance, then that really is a paradigm shift. And so guidance is like the Pediatric Clin Pharm guidance. We're putting it back out in draft form again, I guess. Right at this point, I say that's fine. The science is changing. So why should we finalize it? Which means we probably wouldn't have an update for 10 years. Why should we finalize it? The science is changing. Well, let's keep updating the guidance until we get things right.
Yeah, and I think if we look at other areas of toxicology with more simplistic endpoints, where we've been able to, have validated models now that have gotten us away from the traditional approaches for corrosive testing and other types of erosion testing that we did previously for industrial chemicals, where we can do it all using some of these in vitro models, and they've been validated with numerous chemicals. I think, over time we'll be able to get there with some of these NAMs as well. And it might be a multimodal approach with modeling and with, targeted in vitro studies as well. So.
Final question today.
It's not really a question versus a plea. When we talk about efficiency, rather than waiting for real world data to come in, which we've all discussed is, not as efficient, and then we have to go back and find the mom who maybe had an incident, why not support some universal registries? In worlds where, in their hand on their phone, moms can tell us everything about all the drugs they were on before, during, after. We could do the long-term follow-up. And the best in a clinical trial, the ones that I've run, you're lucky if you get an accidental pregnancy, you follow it at least to the pregnancy. You don't do the long-term follow-up after birth unless, again, it's required, and it often isn't. That's the data we really need. Why go hunting for the mom? Let's incentivize and get them right at the get-go. You're pregnant, be part of a registry.
Just one quick comment on that, if I could. So that reminds me that FDA is really interested in understanding how we can more efficiently, more effectively get that information from pregnant individuals through registries, which we still, okay, this is Lynne Yao speaking, not FDA speaking. I still think that the pregnancy registry is our best hope to collect really quality pregnancy safety information. There, I've said it. Now, we're looking for different alternatives to that registry, but FDA is hoping to convene a workshop sometime in 2025 on improvements to pregnancy registries. The successful design and completion, conduct and completion of pregnancy registries. And before I forget, that also reminds me that another place where we're really thinking, and it really goes to what Lauren was talking about today, is how do we really understand the clinical relevance of transplacental transfer of drugs? So we are holding a workshop in July 11th and 12th, mark your calendars, to talk about how can we better understand transplacental transfer of drugs
and what that means to the infant. What kind of data do we need to collect? What kind of data do we already have?
I would like to see /decentralized product. We do everything through social media, everything is remote, and power that we can have out of that to appeal to the masses and see if anything we can do a standard.
Thank you so much.